All right, so that's a lot of variation from independent assortment. But we actually have to back up a step to get to crossing over. And that's when the homologous pairs are together in prophase 1 and they swap bits of DNA. So it just adds to the genetic shuffling or genetic recombination, making sure everybody is not just different, but really different. Let's take a look. There's one diagram showing it where you got these homologous pairs and they come together and overlap in a process we call synapsis. Then there's the crossing over where they swap bits of DNA and then they look like something new. We call these recombinant chromatids because they've recombined their genetic material. Let's look at what that would look like in action. So here we have homologous chromosomes. There's synapsis. And then they actually swap bits of DNA. Sorry for the hokey sound effects. But they swap them, and then they start getting pulled apart later on in meiosis. And this creates new chromosomes, right? We did just have either blue or orange, but now we have a blue with a little orange and an orange with a little blue. These are 4 unique chromosomes instead of just 2. So this at least doubles the genetic recombination of independent assortment. Knowing that genes travel together on a single chromosome, here I've symbolized genes A through G, we know that they're linked together physically, right? That this is all one big piece of DNA. And so geneticists studied this for a while to figure out how close together are these genes? How close together are genes A and F, for example? And that was kind of a result of how often they split up by crossing over. So, for example, if crossing over split these 2 chromosomes here, then we would get this combination as one of those chromosomes in the end. However, if it's split here and here, then we would get this combination. And by basically running generation after generation, people could figure out that genes A and B were closer together because they didn't split up very often than gene A and F were because they split up more frequently. So genes that are closer together are more tightly linked, and you seldom see those traits appearing separate from one another. Well, crossing over is kind of a crazy and risky process, isn't it? Since when we inherit chromosomes, it's basically inheriting our genetic heritage, right? I mean, that's kind of the idea. You're getting your ancestors' DNA, if you want to think about it that way. And if, like most people, you have a bit of a blended background of ancestry, let's say you have an Irish side and a Mexican side, crossing over basically makes Irish-Mexican chromosomes by mixing up those pieces. Kind of interesting that way. Well, let's move on to meiosis II. There's not a whole lot to say about meiosis II because it's a lot like mitosis, almost the same thing. What do you mean meiosis and mitosis are the same? I thought they were different. Well, what happens to sister chromatids, for example, during mitosis? I think they separate? Right. And that's what happens here. Let's take a look. Here we have an expanded version of meiosis II. Notice we're starting prophase 2 with sister chromatids, but not homologous chromosomes. They get attached to microtubules from each side on that spindle apparatus and pulled apart. Now we have 4 total cells. But we started this one with 2 chromosomes and we ended with 2 chromosomes per cell. So this didn't change the actual chromosome number. Well, let's wrap up by talking about sperm and egg production. Not surprisingly, it happens differently in males and females. Meiosis does. And that's the whole context for all this, right? We're making sperm and eggs, or if your plant's pollen and ovules. Sperm production is all about quantity, making lots and lots of gametes. Egg production a lot slower because the female's also going to be the one that carries the resulting offspring, and so not as much about quantity there. Let's take a look. Sperm production, or spermatogenesis, obviously happens in the testes of males, and each testis is composed of about 300 meters of tubes, these seminiferous tubules. And in those, there are cells lining the outer part that divide by mitosis to produce new cells, which then divide by meiosis and produce the sperm, which mature. You can see some of the resulting tails on the inside here. And so that happens continually. But I wanted to address that issue first. You had this cell along the outside, and it divided by mitosis. One of those cells kind of remain there, and the other one then starts meiosis. And the purpose for that is so you don't run out of cells. If you just took a cell divided by meiosis and you got your 4 sperm cells, like you see down here, well, you'd be done. You'd have no more cells to divide. So this continual renewal process allows continual production of sperm. And this is enormous. This can be up to 400 million sperm every day. This starts during puberty and goes until a male is well on into his adult years, going through meiosis, producing sperm. And the only thing that each of these sperm pass on is 23 chromosomes, and they are tightly packed in the head of the sperm. This is a bunch of mitochondria to power the flagella to get them to swim to the egg. In females, egg production, or oogenesis, produces oocytes that result in a mature ovum ready to be fertilized by the sperm, or the egg cell down here. A couple of key differences. 1, quantity, and 2, the timing of this. It's really strange. And so it starts with mitosis, just like in the males, and everything above this line is before birth. So we start meiosis before the female is even born, but then stop in prophase. So to help us think about that, let's look at the whole process of meiosis laid out. So women are born with all the eggs that will ever be possibly fertilized stopped in prophase 1. And then after puberty, then only one of those at a time is going to resume meiosis, and that stops at metaphase 2. And so basically we're going to go from prophase 1 to metaphase 2 as part of the process of ovulation during the menstrual cycle. All right, but it just stops there, and it's floating around in the fallopian tube, or the oviduct, waiting for a sperm to penetrate. And once it does, after the sperm actually enters the egg, and this is prior to fertilization, then it finishes. And so that's when the sperm actually gets into the egg, it finishes this last phase of meiosis, then fertilization can happen. Another kind of interesting thing, if we look down here, that happens is the production of these polar bodies. And that happens in this first round of meiosis and the second round of meiosis both. And the reason why is you have this big cell and it divides. The egg produces or provides the baby with all the cytoplasm. So we want a really uneven division. Here we want 23 chromosomes, but lots of cytoplasm. And over here, we just want 23 chromosomes and as little cytoplasm as possible. That's the polar body. It ends up dissolving and not being used for anything. In the second division, exactly the same thing happens. Little polar body, big ovum, and that's fertilized and can produce a viable zygote. Really, really different and complicated. But this shows how intricate the human reproduction process is. Well, there's meiosis, and it's an important part of sexual reproduction and producing variation in the next generation, so that no two individuals are alike. And if you look at a really big picture, so that natural selection has some variation to select upon which combinations are good and which ones don't work as well. It's kind of an interesting challenge question sometimes I think of. Could you have identical twins born, let's say, 3 years apart? Well, we know identical twins are possible. Could you have them born 3 years apart? Could you get the same sperm and the same egg produced and those combine again? It's possible based on what we just talked about, but the probabilities are so astronomical that I don't think it would really ever happen. Maybe if human beings continue for another few billion years, there's a chance of it, but probably not. Well, in terms of the whole life cycle and sexual reproduction, meiosis is just half the story. The other half is fertilization. Which sperm fertilizes which egg? And the idea that we have two parents and that we are half of each of them, genetically speaking, creates a lot of interesting variation. Until next time, celebrate you being you. There's only one of you. That's all there will ever be.